So let's not delay this any further. Sorry about all of these issues, and thank you very much for this opportunity to present the results of the work that we've been carrying out on protogadrin 19. And because I am the last speaker today, I'm going to keep my introduction very, very simple. I just want to point out that protogadrin 19 seems to be a protein with multiple functions. It's obvious that there are adhesive properties that are mediated by the extracellular domain. We've heard today that protogadrin 19 interacts homophilically with protogadrin 19, but also with other members of the cadherin superfamily, such as N-cadherin or other protogadherins. There are also interactions mediated by the intracellular domain, which link it to proteins such as the GABA receptor, but also to the actin cytoskeleton through the wave regulatory complex. But the part that we were more interested in was triggered because of this report a couple of years ago by the group of Joseph Goetz, showing that protogadrin 19 could interact with a nuclear protein, which was at that point a bit surprising because protogadrin 19 is supposed to be at the plasma membrane and not in the nucleus. So based on information available for other members of the cadherin superfamily, such as E-cadherin, N-cadherin, or some cluster protogadherins that are processed, we hypothesized at that point that protogadrin 19 might be proteolytically processed, and the idea would be that protogadrin 19 sits at the cell membrane, and then a protease will act as a sheddase and perform a first cut that will liberate the extracellular domain, and it will leave the intracellular domain still attached to the membrane. And then a second protease will come and perform a second cut, and that will then release the intracellular domain into the cytoplasm. And because that intracellular domain contains nuclear localization signals, it can translocate into the nucleus and possibly affect gene expression. And indeed, this model was published last year by the group from Silvia Bassani, where they were investigating the processing of protogadrin 19 in hippocampal neurons. So what I will be focusing on today is very similar work that we've been generating, but in a different brain region, which is the cortex, which is the area that we are more interested in. So here is a little schematic of the protogadrin 19 protein, just to show what to expect in the Western blots that I will be showing. And if indeed there is this first cut happening here close to the plasma membrane, we expect a fragment that we call CTF1 that is more or less 55 kilodaltons in size, give or take, depending on whether exon 2 is present or not. And then the second cut that liberates the intracellular domain should generate a slightly smaller fragment that is 5 to 10 kilodaltons smaller. So based on what was published from other from the processing of other protogadherins, we figured that ADAM10 was a very good candidate to be acting as a sheddase for protogadrin 19 because it has been shown to cut or to make that first cut for all other members of the cadherin superfamily that have been investigated. So we started our analysis not with neurons yet, but with a different type of cell, which are mouse embryonic fibroblasts. And we did that for two reasons. First, because we could get hold of ADAM10 knockout MEFs, which is a very powerful tool to investigate the involvement of this protease, but also because MEFs express endogenous protogadrin 19, which makes our life much easier because we don't have to transfect it. So we took these ADAM10 wild-type and knockout MEFs, and we treated them with ionomycin. And ionomycin is a calcium ionophore that is that it has been shown to trigger processing of other proteins. So as you can see here, when we treat with ionomycin in the ADAM10 wild-type MEFs, we can see a strong increase in the intensity of CTF1, which is indicative of processing happening. This effect is stronger initially, and then with more prolonged treatments, it kind of goes down. So apparently the processing happens at the beginning, and then that's it. And then when we treat with an ADAM10 specific inhibitor, what we see is that this increase in the intensity of CTF1 is abolished. So it really seems to be telling us that ADAM10 is involved in the processing. However, I want to point out that there is still a certain amount of processing going on, which could potentially mean that the inhibition is not complete. However, we then looked into our ADAM10 knockout MEFs, and we have done Western blot analysis with these cells, and we cannot see any residual expression of ADAM10 at all. So what we see here is that the effect of the ionomycin treatment is gone, and we don't see any significant increase in the intensity of CTF1 when we treat with ionomycin. However, as you can see, there is still processing going on in the complete absence of ADAM10. So this was a first indication that yes, ADAM10 can cut protocadherin 19, but it's definitely not the only protease that can do this. Then we wanted to check this in a more relevant system. So we wanted to start looking into neuronal cells. And for that, we used ES-cell-derived cortical-like neurons that we can generate from ES cells, and that provide us with a relatively homogeneous type of neuronal population to work with. And to our surprise, when we treated them with our ADAM10 inhibitor, we couldn't see any difference to the untreated cells. So we treat with ionomycin, we see this increase in the intensity of CTF1, but then if we've treated with the inhibitor, there is no change whatsoever. So first thing you think, okay, my treatment didn't work this time for whatever reason. However, we've done this several times, and we've taken those same lysates and we have blotted them for cadherin-2, which is a known substrate for ADAM10. And here we can see this very strong effect of the inhibitor, where we completely abolished the processing of cadherin-2. So what these experiments are telling us is that ADAM10 is present in these neurons and it can cut cadherin-2, but it's not cutting protocadherin 19. So even though in MEVs we see this processing going on, in these cortical-like neurons, ADAM10 doesn't seem to be involved in the processing. So we wanted to see what else could be involved. So we tested two more protease inhibitors. The first one was verubetastat, or MK-8931, and this is an inhibitor for base 1 and 2 proteases. And when we used this inhibitor, we couldn't see any differences compared with ionomycin alone, so we can rule out the involvement of these two proteases. However, then we used batimastat, or BB94, and this is a potent broad-spectrum metalloproteinase or matrix metalloproteinase inhibitor, which inhibits MMP1, 2, 9, 7, but also inhibits other proteases such as ADAM17, I think ADAM10 as well. And here we could definitely see an effect. So this increase in the intensity of CTF1 is completely abolished if we apply this inhibitor. So take-home message is that ADAM10 might be processing protocadherin 19 in certain cell types, but not in others, and there are other proteases that are involved. And in a way, I think this shows that processing of protocadherin 19 might be cell type dependent, stimulus dependent, or a combination of two. And the fact that there are more proteases that can be involved here, I think, speaks towards how biologically relevant this processing is. Then we looked into the second processing step, and again the obvious candidate was gamma secretase, because again it has been shown to cleave all other cadherins that have been shown to be processed. So we used DAPT, which is a specific inhibitor for gamma secretase, and we could see that when we treat neurons with this inhibitor, we see an accumulation of CTF1, and because CTF1 is the substrate that is used by gamma secretase to further process, if we see an accumulation, it means that gamma secretase is not being able to process it further. So this speaks in favor of gamma secretase being involved. However, again, we can still see the appearance of CTF2, again pointing to the fact that other proteases might be involved as well. So we took a relatively wild guess and tested calpain. So we used calpeptin, which is an inhibitor for calpain. Calpain is a calcium-dependent protease that has been shown to process E and N cadherin as well. So when we treated our neurons with calpeptin, although our results are not completely consistent yet, but what we can see is that although there is less accumulation of CTF1, which is not significant compared to the control, we can see a reduction in the formation of CTF2. So I'm not going to say that calpain is definitely involved in the processing, but it seems to be able to do something. And definitely, I don't think that gamma secretase is the only protease that is generating the second CAR. So the processing of protocadherin 19, I think it's more complex than just ADAM10 and gamma secretase. Another interesting point about the processing, which has also been shown in hippocampal cells, is that it is activity dependent in neurons. So we did this with the in vitro 12 neurons, which are mature enough to respond to NMDA. And when we treated them with NMDA or with KCL, which I am not showing here, we could see this huge increase in the intensity of CTF2, which seems to be the main product that is being produced in neurons. And then if we treat with MK801, which is an NMDA receptor antagonist, that effect is gone. Of course, this is all nice, but it's all done in vitro. So we wanted to see or get some kind of indication of whether all of this is happening in vivo as well. And the other interesting bit with this part of information here is that we would expect to have more processing of protogadrin 19 in neuronal cells that have activity than in other cells in the brain that express protogadrin 19 but are not electrically active. So we took advantage of the fact that very early on during development, protogadrin 19 is expressed in progenitors, and progenitors are not electrically active. So what we did was just take lysates from E11 cortex here, and then from P10 cortex and hippocampus, from wild-type and knockout animals, and we just did a Western blot to see what we could find. And as you can see here, we can nicely detect full-length protogadrin 19 to roughly the same extent in terms of how much of it is being expressed. However, if you compare down here where the process fragments are, we can very nicely see processing happening in the P10 cortex and hippocampus, but we can barely detect anything in the progenitors. So this, to us, is an indication that processing of protogadrin 19 in neuronal tissue is mostly probably activity dependent, and that there is not a very high level of basal processing, as we can see, for example, in the mouse embryonic fibroblast. So what happens to this piece of protein that is released after processing? Well, it has already been shown that it goes to the nucleus, but we wanted, all of this has been mainly done in culture, and we wanted to see how it is in vivo. So for this, what we did was compare the subcellular localization of full-length protogadrin 19 and of the cytoplasmic domain in cortical neurons in the cortex. To look at the full-length protein subcellular localization, we took advantage of a combination of CRISPR-Cas9 and in utero electroporation, and we tagged the endogenous protogadrin 19 with an HA tag. And here you have an example of a neuron that was successfully tagged. This is stained with anti-HA, and you can see that the full-length protein, and this is not, I want to stress out, this is not overexpression, this is the endogenous protein being produced. It seems to be at the membrane and probably also being produced and transported, but it doesn't seem to be localized to the nucleus at all. However, if we use in utero electroporation to express the cytoplasmic domain, again tagged with HA, we can see that it definitely goes into the nucleus. So this is also something that is going on in vivo. But again, this experiment with the intracellular domain, this is a piece of protein that we are expressing. This is not something that has been generated in the neurons. So we wanted to see how the processing is going on. So we went back to cells, we took HeLa cells and transfected them with full-length protogadrin 19 and treated them with ionomycin. And then we quantified the fluorescence intensity in the nucleus versus cytoplasm after 20-minute treatment. And what we can see is that there is a significant increase in the amount of fluorescence in the nucleus after treatment with ionomycin, because we are comparing two cells that have been transfected with the exact same construct, but one has been treated and one has not been treated. This is telling us that there is processing going on. And of course, we assume that it is the nuclear localization signal in the cytoplasmic domain that is making this fragment go to the nucleus. So we express wild-type intracellular domain, and we can see very, very strong nuclear fluorescence. And if we mutate part of the nuclear localization signal, there is a significant decrease. However, I want to point out that there is still some protein going into the nucleus. So there's probably other fragments within this, within the intracellular domain that can help bring the protein into the nucleus. Nuclear localization signals work by interacting with a family of proteins called importins that will identify that nuclear localization signal and then bind the protein and carry it through the nuclear pores into the nucleus. And we had originally found, and I presented this last time here, that Protocaterin 19 interacts with KPNA1, which is one such importin. It's also called importin subunit 5. And that we could see a stronger interaction when we express the intracellular domain alone than when we express the full-length protein. But again, this is not telling us whether this is happening in vivo, because this is just overexpression of proteins in a cellular system. So what we've checked now is whether, something weird happened there. Sorry, this was a presentation not done in PowerPoint and then converted into PowerPoint. Anyway, bear with me. What we've been able to show is that if we take P10 lysates from the cortex and we perform immunoprecipitation with protocaterin 19, which would be this first lane here, if we blot, we can detect KPNA1. And if we do the immunoprecipitation with anti-KPNA1, which would be this lane here, we have a very strong co-precipitation of this protein, but we can also detect protocaterin 19. So this is an interaction that is happening in vivo as well. So I think with this data, we can be sure that processing is happening in the cortex. It's something that is not just triggered artificially in vitro. This is going on, and the fragment goes into the nucleus. So what happens then? Does it have any influence or any effect on gene expression? So to look at that, we used ES cells that we modified to express, in addition to regular full-length protocaterin 19, we put this construct into the ROSA26 locus to express the intracellular domain fused to HA. And here you have our subclones where 1.2 and 1.18 are positive, so we can detect a stronger expression of the intracellular domain, and we can also detect it with HA. So we know that it is our fragment that is being overexpressed. So the first question, of course, was can we take these ES cells and differentiate them into neurons, or does this overexpression have an effect or an impact on that process? But luckily for us, we could. So they would proliferate normally. We could differentiate them into neurons. So what we did was take these differentiated neurons at the in vitro 8, which are relatively immature neurons, and then at the in vitro 12, which are more mature. And we use them to perform bulk RNA-seq. And down here, you can see that our overexpressing cells are truly expressing more of the cytoplasmic domain. So this is exon 1, and the counts, the reads for the wild type and the overexpressing cell lines are the same, but then all of these exons that code for the intracellular domain show much higher counts in the overexpressing cell lines than in the wild type. So what did we find with this? Well, we found that there are many changes across the transcriptional landscape, both at the in vitro 8 and 12. We see roughly the same number of genes that are up and down regulated at both ages. And when we compare the genes that are affected, we can see that about half of the cells are common between the two ages. And interestingly, if they are overexpressed at the in vitro 8, they are overexpressed at the in vitro 12. And if they are downregulated at the in vitro 8, they are downregulated at the in vitro 12. And this is telling us that there seems to be a continuous regulation by this protocaterin 19 intracellular domain across the development of these neurons. So of course, we wanted to know more about the type of genes that are affected. So we performed GO-term analysis, and here you have the results using gProfiler. These are the top 15 terms for biological process, and these are the top 15 terms for cellular compartment. And you can see that we can find terms like nervous system development, cell differentiation, neuron differentiation, and also neurogenesis and generation of neurons. When we look at the cellular compartment, it gets very interesting because we see a lot of terms here in green relating to the synapse or to neuronal projection, so neuronal projection, synapse, postsynaptic membrane, etc., and many other terms that are also referring to the membrane. So there seems to be something going on with the regulation of membrane genes. And here you have an example of some of these. These that I am showing here are the differentially expressed genes that were specifically linked to neuronal differentiation at the in vitro 8, and we see a lot of transcription factors. Many of them are homeodomain or zinc finger transcription factors, but we also find many membrane proteins And I think an interesting one here could be, for example, FreeCell10 in terms of Wnt signaling, or LRP4. And we also find other proteins, including many secreted proteins as well. So it seems to have a broad impact over many genes that are involved in neuronal differentiation. So of course the question is, this is done with ESL-derived neurons, what is happening in vivo? What happens to neurons in their normal context if we overexpress this intracellular domain? So we did in utero electroporation experiments where we electroporated either a control plasmid expressing just GFP or our overexpression plasmid with the protogadrin 19 intracellular domain and GFP. We electroporated at the 15.5 to target upper layers in the cortex because we know that this type of neurons express protogadrin 19 normally. And then we waited until P60 and then did the analysis at that age, so roughly 2 months after electroporation. So because we had these terms relating to neurogenesis, we wanted to see whether there was any influence there. There doesn't seem to be. Electroporation like this is not the best way of looking at that because of different efficiency of electroporation between animals, but by looking at several animals, what we have seen is that there is no difference whatsoever in the average number of electroporated cells and no difference in cortical thickness. So although this is not 100% ruling out an effect on neurogenesis, there doesn't seem to be any major effects. Then we also looked at migration because although I've only shown the top 15 Go terms, migration was also one of the terms. And you can see here that the neurons that express the intracellular domain of protogadrin 19, they seem to settle on a slightly wider area and maybe slightly lower, but so far with our data we don't see a statistical difference. So we cannot really claim that there is an effect on migration. Another couple of terms that show up were terms related to axonal guidance and axonogenesis. So we had a look at in our electroporated brains, and I think you will need to trust me when I tell you that what we have seen are no differences. So we can see the arborization of the upper layer neurons in layer 5. We can see it here. It's weaker, but this has to do with fluorescence intensity and plasmid architecture. We can see, we expect these upper layer neurons to project through the corpus callosum, and this is exactly what we see. We don't see any weird axons in the striatum or anywhere else where they would not belong. And we can follow them to the contralateral hemisphere where the signal is very weak, but if you squint a little, probably you might be able to see that there are axonal projections reaching the contralateral hemisphere in an equivalent position as in the wild type. So next, we looked at neuronal morphology. So we took these electroporated neurons and we traced them. And of course, it's difficult to know because this is layer 2-3, so there are different types of neurons, so we've tried to be as comprehensive as possible. And again, we've looked at, for example, width of the dendritic arbor, of the apical and the basal one, no differences there. We've looked at the sum of lengths of dendrites of the different orders, we see no differences, and you can see there the Scholl analysis, where we can see that there doesn't seem to be any differences. So at this point, we were pretty frustrated, obviously, thinking, well, there doesn't seem to be any effect whatsoever of overexpressing this intracellular domain. But there is one more thing that we can analyze. And when we think about all of these terms in the cellular compartment analysis relating to synapses, nothing of the analysis that we've done, none of the analysis that we've done so far is looking really at that. So we decided to look at dendritic spines, because dendritic spines is where most of the excitatory synapses are happening in the cortex, at least. And then finally, we found our result. So what we were able to see was that in our neurons that are overexpressing protogadrin 19 intracellular domain, we see a roughly 40% reduction in the number of spines. And this affects in a very similar way both apical and basal dendritic spines. And we are now waiting to do our last experiment to verify that this is a real phenotype and not due to differences in fluorescence intensity. And what we think, well, we don't know what is the mechanism behind this, but one possibility would be the XLR genes. So there is this paper from Marta Nieto's lab that showed a while ago that in CAX2 mutants, there are differences in spine density. And what they found was that the expression of these XLR genes was increased in the CAX2 knockout animals, and that this led to a reduction of 40 to 50% in the number of spines. And what we have seen is that in our ES-cell-derived neurons that are overexpressing the intracellular domain of protogadrin 19, we also see a pretty significant increase in the expression of these XLR genes. These are relatively unexplored genes. They're a family that are located on the X chromosome. The conservation between mice and human is not that great, but because they've already been shown to be involved in spine density, we believe that maybe that could be one potential mechanism. So to finish, I just wanted to present this potential model where protogadrin 19, at least in cortical neurons, could potentially act as an activity sensor, so that if there is a lot of activity going on, in principle, one would expect to have a lot of processing happening and a lot of intracellular domain being produced. That intracellular domain can then go to the nucleus, and according to our data, that is going to create a signal to reduce the number of spines. So in principle, at least in cortical neurons, we could have a situation where protogadrin 19, one of its many functions could be to detect neuronal activity and help with plasticity or homeostasis so that neurons can deal with increased activity by maybe downregulating the number of spines. So with this, I just want to finish and thank the people who've been involved. And the main person who carried this work forward was my former PhD student, Sylvia Newbold. She did most of the work, but also with help from Ian Fox and Dr. Cristina Linares Venadero. We've also had help from Dr. Cristina Gil-Sanz and Jaime Fabra at the University of Valencia, and also thanks to my funders. And thank you for listening. Layer 5 neurons?
Yes, the layer 5 neurons, so something around 12.5 or whatever?
Yes, so we had to decide which age to go for. The problem with trying to target layer 5 is that for that you need to electroporate at E13. And when you electroporate at E13, you don't just get layer 5 neurons, you get layer 6, you get layer 5, and you get some layer 4. So the breadth of neurons that you get electroporated is much bigger. The other problem is when you then want to trace them, layer 5 neurons are much more difficult to trace because you need to be able to follow them through a much longer distance with their apical dendrites. So this is why we initially decided to go for layer 2-3 neurons. Yeah. Logistical reasons, and because they also express protogadrin 19, so we would expect them to be a good population.
Quick question. Did you see any changes in ion channels?
In the RNA-seq,
yeah.
yes, we see changes in some ion channels, voltage-gated calcium channels, if I remember correctly, and others. We see changes in neurotransmitter receptors. And, well, well, with 200 genes plus altered, but yeah, there are some changes in ion channels. Yeah. You had another question?
Well, it was a simple question, but when you count the number of spines, have you considered the morphology as well, so that you know, like more informative?
Yes, we have, and we have an analysis kind of going on with that. But so far, our data don't seem to show any differences.
We published a paper some years ago, and we performed this study on brain MRI, measuring the cortical thickness in some patients with PCDH19 mutation, and we showed that there was some difference in cortical thickness in some of these patients. So we hypothesized actually that, I don't know, I mean, we don't know if there was because of the seizures or because of the PCDH19 mutation. So because, of course, we performed the MRI later on in life and the patient had already had some seizures. So, but there seems to be some difference in cortical thickness in the sort of the amygdala and the area and the, yeah, that's, because, well, that's where the seizures are coming from anyway.
Yeah. Well,
Oh,
here it would be a bit, so when you electroporate, you don't target all neurons. So it's more of a mosaic overexpression. But we've been looking at the protogadrine knockout animal, and we did a thorough characterization. We saw no differences in cortical thickness, but of course these are mice and not human, and there's always a certain degree of difference there. We also looked much earlier, we looked at P10. So if you looked at adult patients, maybe that could also explain why we don't see any differences. But in this particular case here, cortical thickness was kind of an attempt to check whether there would be any non-cell autonomous effect, because the amount of neurons that we target compared to all of the neurons that are there, it's relatively small. The other thing is, of course, we are electroporating at E15.5, which means that all of the deep layer neurons have already been generated. So you would not expect to be altering them in any way. And we would only be targeting the upper layer neurons. So I guess that's a combination of factors.
So it's just more of a technical question, maybe I missed it. The way you showed where we said NPP CDH19 antibody or was TAG?
Which one?
I don't know when you had the 180, 130 kilodalton, maybe this one?
This is all,
one next, or even
this is protogadrine 19 antibody.
So those two bands, so the antibody is against C-terminal.
Yes. Yeah.
So how come you see the two bands for the full length? They are surely that's a double.
So we see two bands for the full length. We've done treatment with PNGAs because we thought, oh, maybe it's glycosylation. It's not, because when you treat with PNGAs, you see the exact same two bands, but lower. So what I think that these two bands are is plus and minus exon 2.
That was my question, because that's where the nuclear localization sequence.
Yes and no, it's half of it. So the bipartite.
But you lose it if you take the exon 2 out.
I think what you do is convert a bipartite localization signal into a monopartite localization signal. But the other thing is, here we are just detecting the proteins. We're not looking whether they go into the nucleus or not, because this is just total lysate. So in principle, the processing could still be going on. If the nuclear localization signal, or if the part of the nuclear localization signal that is in exon 2 is relevant, then it could be that after the processing, you get two products. One with exon 2 that goes to the nucleus, one without exon 2 that might or might not go to the nucleus. But considering that when you mutate the nuclear localization signal, you still see some nuclear localization, I think that it's not only that region that it's important for nuclear localization.
So maybe other proteins which may drive it. Because the exon 2 is very small.
Yes.
I'm intrigued by those two bands.
Well, I mean, what I can tell you is that apart from the isoform lacking exon 2, there's also an isoform lacking exons 3, 4, and 5. No, sorry, 2, 3, and 4. I don't think it's a very common one, but when I first amplified protogadrine 19 with primers at both ends, I got the version without exon 2, and I got a version without exons 2, 3, and 4 that just read through from exon 1 straight into exon 5. Which is interesting because the conserved motifs are in exon 5 and 6. So